Good afternoon, everybody. I'm so delighted to see all of you here. I'm Janine Clayton, the Director of the Office of Research on Women's Health, and it's really my pleasure to introduce today's WALS lecturer, Dr. Yang Dan. Dr. Dan is a professor of neurobiology at the University of California at Berkeley and an investigator with the Howard Hughes Medical Institute. Following undergraduate studies in physics at Beijing University, she completed a Ph.D. in neurobiology at Columbia University in Muming Pu's laboratory. She was a Grass Fellow at Woods Hole, then moved on to postdoc work at Rockefeller and Harvard Medical School in R. Clay Reid's laboratory. Her lab uses electrophysiology, imaging, optogenetics, and computational techniques to study functions of the mammalian brain. They have studied microcircuits underlying visual cortical computation and mechanisms for cortical plasticity at multiple levels, from synapse to perception. Her lab has made important contributions in understanding the synaptic bases of functional plasticities, in particular linking spike timing dependent plasticity at the synaptic level to visual system function. Her recent work has revealed the mechanisms by which brainstem, hypothalamus, and basal forebrain circuits exert powerful control over sleep-wake brain states. She has an outstanding record of mentorship with numerous trainees, going on to highly successful tenure-track positions. This afternoon, she will speak to us regarding neural circuits controlling sleep. Please join me in welcoming Dr. Yang Dan.
So can you hear me, the microphone? Thank you. It's really a great pleasure for me to be here, and I'm really greatly honored to be given the opportunity to present in this prestigious lecture series. And thank you so much for coming here in this snowy day. So what I'd like to do today is to tell you about our studies over the last 5 years on the neural circuits controlling sleep. Mammals and birds have 3 distinct brain states: wakefulness, non-REM sleep, and REM sleep that can be clearly distinguished based on EEG and EMG. Obviously, during wakefulness, we have vivid sensation of the external events. The EEG is showing this sort of the so-called desynchronized pattern, meaning that it's dominated by small amplitude and fast activity. The EMG shows a high muscle tone. And during non-REM sleep, which is also called slow wave sleep, the EEG is dominated by these large amplitude slow waves. The EMG shows reduced muscle tone, and we have dull or absence of sensation. Now, REM sleep, rapid eye movement sleep, was discovered only about 60 years ago in the 1950s. It's also called paradoxical sleep. Paradoxical in the sense that the EEG is actually very similar to the wakefulness EEG. But the EMG shows a complete lack of muscle tone. So this is the so-called muscle atonia or REM paralysis. And this is the state when we have vivid dreams. And so the idea is that the desynchronized EEG, the wake-like EEG, is what allows us to have this sort of vivid mental experience. But the muscle paralysis can prevent us from acting out our dreams. So the question that we have been focusing on is what mechanisms control sleep and wakefulness. Now, over the past many decades, studies from many labs around the world have identified multiple brain regions that are important, including brainstem, hypothalamus, and basal forebrain. So these include many of the well-known neuromodulatory circuits. For example, the monoamines, including noradrenaline in the locus coeruleus, histamine in the TMN, and dopamine and serotonin and so forth, acetylcholine in the brainstem and basal forebrain, and some neuropeptides in the hypothalamus, including orexin and MCH. Now, as it turns out that most of these well-known neuromodulatory systems, as far as sleep-wake regulation is concerned, their function is to wake up the brain, with very few exceptions. Now, there are also many conventional glutamatergic and GABAergic neurons in these brain areas. And as it turns out that if you just randomly pick some of these neurons to activate, then more than 9 out of 10 cases, you find that these neurons can promote wakefulness rather than triggering sleep. Now, I didn't know this 5 years ago when I got into sleep research, but now I know this very well. Now, just to give you an idea of what I'm talking about, So here is a video of activating some glutamatergic neurons in the hypothalamus. Now every time you see the blue square turning on, it means that we turn on the laser. And you can see that this mouse is going crazy in this trial. And even after we turn off the laser, he was still going crazy. Now this is the second trial. He was actually in non-REM sleep, but the moment that you turn on the laser, they start running around in a pretty agitated way. So this is pretty much what you see most of the time if you just optogenetically activate some of the glutamatergic or GABAergic neurons in these brain regions. In other words, it's really easy to find neurons that can wake up the brain. It's much harder to find neurons that can trigger sleep. So the obsession of my lab recently has been to try to search for the neurons that can promote sleep with the hope that once we understand the sleep circuit, then we can perhaps come up with ways to improve sleep because insomnia is a very common sort of problem in the modern society. So what I'm going to do today is to first focus on this region, the brainstem, and then in the second part, I'll move to the more anterior part of this circuit. So the brainstem has been traditionally closely linked to REM sleep. And this was figured out using the transection studies. So these studies were pioneered by Juve in the 1960s and later followed by many investigators. But this is a diagram from a review paper by Jerry Siegel. Basically what people found is that if you make a cut or transection at level A or B, so this is a sort of a sagittal view of the brain showing the brainstem. So if you make a cut at A or B, then in the forebrain region, anterior to the cut, you see signs of non-REM and possibly wake, but there's no sign of REM sleep. But you would still see once in a while REM atonia, right? So this loss of muscle tone. But if you make a cut at C, then you see normal REM in the forebrain, but there's no spinally mediated atonia. So these experiments, so this is actually summarizing many, many lesion studies from many labs. But these studies basically suggest that the region between A, B, and C, which includes the pons and the medulla, these regions are important for REM sleep. And people have also made some cuts in D and E to show that the communication between the pons and the medulla is important. So but of course, those brain regions are also involved in many other functions. And to identify the specific neurons that are important for sleep, the traditional method has been one of the methods is to use so-called CFOS staining. So what you do is you first do a REM sleep deprivation. So there are different ways of doing it, but the general idea is that every time when the animal goes into REM sleep, you wake them up. You either poke them or use some other higher tech methods. But then you do this for several hours, and then you leave the animal alone. And that usually causes a REM sleep rebound because both REM and non-REM sleep are heavily regulated homeostatically. So after deprivation, you leave them alone, they spend more time in REM sleep. And you leave them alone for about 2 hours. That's enough time for the immediate early gene CFOS to be expressed. And then you sacrifice the animal, you stain for FOS. So this is a study done by the Lupi group in Lyon in France. So this is a coronal section cut at about this level. So you can see that it's very posterior in the brainstem, actually in the medulla. And so in this picture, you see these black dots. So these are actually the CFOS positive neurons. And they also figured out that a lot of these CFOS positive neurons are actually GABAergic neurons, so these inhibitory neurons. So, since this is after REM sleep rebound, it suggests that these neurons are REM sleep active. And this is usually the first step that you need to say perhaps these neurons are important for REM sleep. Because if they're not active during REM, it's very hard to imagine that they're important for REM sleep. So, the questions that we asked after being inspired by these results is, first, does the ventral medulla, which I'll abbreviate as VM, does the GABAergic neuron activity cause REM sleep, right? Because the CFOS experiment is actually correlation, but it doesn't mean causality. And second, are these neurons specifically activated during REM? And this is important because even though these neurons are CFOS positive after REM rebound, what the problem here is that Okay, so REM rebound means increase of REM sleep, but normally we only spend about 10% of the total time in REM sleep. So REM rebound causes this 10% to increase to about 40%. It's a fourfold increase. It's very dramatic, except that during those 2 hours, the animal still spends about 60% of the time in other brain states, right? So the fact that these neurons are CFOS positive, we're not absolutely sure that these are specifically active during REM. So the only way to tell that is to actually record from these neurons. And then if these neurons, actually their activity causes REM sleep, what are their downstream targets? And if these neurons are specifically active during REM, then what are their inputs, right? So these are the standard questions that we ask when we try to figure out the neural circuits underlying behavior. So to answer the first question, this is what we did. It's very straightforward and simple. Now we have this magic tool called optogenetics, right? So we inject the AAV virus that expresses channelrhodopsin in this region where they found the CFOS positive neurons in the ventral medulla, in particular this nucleus called LPGI. And the AAV carries the channelrhodopsin and YAP gene, but in a Cre-dependent manner. So if we inject this virus into the GAD Cre animals, then the virus will only be expressed in the GABAergic neurons. And then we insert an optic fiber into this region. So this is a picture of this small region. And you can see the expression of the neurons, and our electrode is about here. So then we turn on the light and see what happens. So here's a video just to give you an idea of what our experiments is like. So again, on the left is the video of the animal. So if he's sitting here, he's probably asleep. And when you see a blue square, that means that we turn on the laser. So this upper panel shows the power spectrum of the EEG. Like I said, EEG is a very important measure for determining the brain state. So if you see a lot of warm color down here at the low frequencies, it means that the brain is in the synchronized state, and that usually indicates it's non-REM sleep. But if you see a very thin band, about 8 hertz, that's a so-called theta band. Usually that means that the animal is in REM sleep. And if you see a reduction of the low frequency, and if you see a high EMG, that means that the animal is awake. So here in this first trial, you see a lot of low frequency activity. He was in non-REM sleep. And you see as soon as we turn on the laser, pretty quickly there's this change of the EEG pattern. And at this point, you start seeing this very thin band. So this is the theta band. And the EMG amplitude stays low. So that means that this animal was in REM sleep. Now here's another trial. Again, he was in non-REM sleep because of this broadband activity in the EEG. Turn on the laser, pretty quickly you see a change, and now you see a very thin theta band. And at this point, you see the increase in the EMG, so he woke up. Anyway, so this is just how we do the experiment. And this is a summary of that particular mouse, but recording over a longer period of time. So here we apply laser in 4 trials. Now the laser was applied for 2 minutes in each trial, and the inter-trial interval is 15 to 25 minutes. It's sort of semi-random, controlled by the computer. Now, so if you don't want to always look at the EEG and EMG, you can just look at this color-coded brain state, which is determined based on EEG and EMG automatically by the computer program. So you can see that in 3 of the 4 trials, right, the laser was turned on when the animal was in non-REM sleep, this gray region, and soon after the laser was turned on, it turned blue, right? So that means that the brain transitioned into REM sleep, right? So that happened here, here, and here. Now, in one of the trials, we turned on the laser when the animal was awake, and you actually saw that nothing happened. This animal stayed awake. So this is a summary of the data from 6 mice, all the trials that we applied. We aligned these trials by the time of laser stimulation, which is this bar here, right? So it's 2 minutes per trial. So, I mean, just sort of eyeballing it, you can see that during the laser period, there's a lot of light blue, right? Indicating he was, most of the time he was in REM sleep. Now, these lines shows the probability of the 3 brain states. So non-REM, right, gray, So before laser, he spends about 60% of the time in non-REM sleep, and this is normal for mice because they're nocturnal, so we do experiments during the day, so this is their average probability of non-REM sleep. Now, as soon as you turn on the laser, this gray line, you see this dramatic drop, almost to zero. And accompanying that, there's this dramatic increase of the light blue line, right? So there's a dramatic increase of REM sleep. Now, there's also a slower increase of the purple line, which is the probability of wakefulness. Now, I just want to say here that actually, we don't think that this is because the laser was triggering wakefulness. We think that this increase is because of the fact that in rodents, after REM sleep, they always wake up. They don't go back to non-REM sleep for some reason. I think that humans can do that, but rodents don't do that. And so because the laser is driving such a high probability of REM, and then after you enter REM, you have nowhere to go. You have to wake up. And that's why there's this increase of wakefulness. And I'll show you the data to support that claim. But back to this data, right? So if we sort the trials and say, let's look at all the trials when we turn on the laser when the animal was in non-REM sleep, right? So we sort a subset of the trials shown here, right? And so you see that among these trials, the probability of triggering REM sleep is about 90%, right? So it's highly, highly efficient. But if you look at the trials where we turn on the laser when the animal was awake, actually we don't see really any REM, right? So the effect of laser really depends on which brain state the animal was in. So in addition to just look at the probability of the three brain states, we also looked at the transition probability. So this is a plot showing the probability of transition from non-REM to REM. And so the way we analyze that is to say, okay, at each time point, right? So this time point is defined relative to the time of laser stimulation. So at each time point, we look at how many trials in which the animal was in non-REM sleep. And then within the next 20 seconds, in how many cases did the animal transition into REM, right? So let's say you have 100 non-REM trials, and within the 20 seconds, 30 of them got into REM, so then the probability would be 30%, right? So that's how we calculated this. And you can see that laser is this shading period. Now, before laser, the transition probability is incredibly low, right? It's maybe 5% or something. But during laser stimulation, there's this very dramatic increase. I think it was 20 to 30-fold increase of the transition probability from non-REM to REM, which is consistent with what we see in the last slide. But now, if you look at the REM to wake transition, actually, if anything, there's a decrease, right? So this is why I'm claiming that even though in the last slide you do see a slow increase of wakefulness, we don't think that this is triggered by laser because the probability from REM to wake is actually decreased by the laser. Now, this is the probability from non-REM to wake. There's actually no significant change, right? So because these are the only two transitions that can cause wakefulness, and one is decreased, the other is no change. So that's why I'm saying laser does not directly trigger wakefulness. It's only promoting REM. Now, the last probability I wanted to show is that the wake to REM transition, it turns out that if you're a normal subject, you actually don't have this direct transition. You have a direct wake to REM transition only if you're narcoleptic, right? So if some of you actually notice that, you got to go see your doctor, right? So that's a pathological condition. Now, as you see, before laser, these mice seem normal. They don't have this transition, and laser does not cause that transition either. So the bottom line is that even though our laser stimulation of these ventromedulla neurons can really promote REM sleep, it's only the normal non-REM to REM transition, but not the pathological wake to REM transition. Okay, so of course, if you do any optogenetic experiments, you have to do your controls. So because you're sticking the optic fiber into the brain, and you turn on the light, who knows, maybe the mouse could see the light. And so the control for that is you express only YFP without channel rhodopsin in these neurons, right? Everything else is the same. You're only skipping channel rhodopsin. And you can see that using the same light stimulation protocol, we're not increasing REM sleep or anything else. So that suggests that this is really due to the activation of those neurons and not just light per se. In addition, if we actually express the virus not in the GABAergic neurons, but in the glutamatergic neurons using the VGLU2 Cree mice, then only 10 seconds of laser stimulation dramatically increases the probability of wakefulness and decrease of both non-REM and REM, right? So this suggests that the effect is not only specific to that area, but also specific to the cell type. So if you activate the wrong cell type, you increase wakefulness. And like I said earlier, promoting wakefulness, those neurons are everywhere. It's really easy to find them. Okay, so that in that experiment is what we call the closed loop stimulation. We basically, the computer is blind to what the mouse was doing. It just turns on the laser once every 15 to 25 minutes. But here is an experiment that we use the closed loop stimulation. So we know that we can trigger REM, right? But we also wanted to know whether we can maintain REM. So if the animal is already into REM sleep, can we prolong the REM sleep? And so for that, we like to leave the transition into REM to the animal herself or himself, right? And then we only turn on the laser after the transition has happened. So what we did here was use a computer to have a real-time monitoring the EEG and EMG. And every time we detect a REM sleep, right, so this is what the animal did on their own, and then we tell the laser, the computer tells the laser to turn on. And this happens about 50% of the times. And the laser will stay on as long as the REM is there. So only when the REM ends, we turn off the laser. So in other words, we want to keep the laser on, right? We don't want to turn off the laser to trigger the end of REM. We just want to keep the laser on and see how long we can promote REM. So here is an example experiment, and you can see that here is a REM period, right? So we didn't turn on the laser because whether you turn on the laser is determined stochastically by the computer. It's 50%. So this trial, we didn't turn on the laser. This trial, we did. These two trials, we didn't turn on the laser. This trial we did, no laser, laser, right? And even just eyeballing it, you can see that in these three trials where we turn on the laser, it seems like the REM was lasting longer, right? And if we summarize all the 6 mice, all the trials, so these are within the subject control, right? Because each animal you have some REM that with no laser, some with laser. So if you compare the REM duration, no laser is about 70 seconds on average, but with laser, it basically doubles the duration. Now, this doesn't happen if we use the EYP control mice, right? So there's no change. Now, when we use this closed loop stimulation, if we activate ARCH or halorhodopsin, which suppress the neural activity, we can actually reduce the REM duration. And again, that doesn't happen with YAP control. So just to summarize our answer to the first question, we showed that activating these VM GABAergic neurons optogenetically can induce the non-REM to REM transition. In our hands, it's 94% efficiency, so it's incredibly high. And also, we can double the duration of REM sleep episode. Now, what about the second question? Are these neurons specifically active during REM? So this is very important because optogenetics is really a technique that has taken the field by storm. Everybody does that because it's fun, right? You turn on the light, usually the animal, you see something happens. But of course, one thing you always have to ask yourself is that whether your effect is physiological, right? Because you don't know what these neurons are naturally doing. So what if you're activating these neurons in a very non-physiological condition, you're causing some effect, it doesn't tell you how the brain works. So we also wanted to know really what's the firing pattern of these neurons when we're not driving them with laser. So of course, the problem here is that in this region, they're not only GABAergic neurons, they're also glutamatergic neurons. In fact, they promote wakefulness. And in addition, there are also serotonin neurons very close by, right? So how do we know that we're recording from a GABAergic neuron? So the way we do that is to, again, tag the neurons with channelrhodopsin, right? So if we use the GAD-CRE mice, then we know that the virus is only expressed in the GABAergic neurons. And then we use the so-called optrode. It consists of an optic fiber surrounded by several bundles of wires for recording. So when we insert this into the brain, we look for neurons that are reliably driven by laser. So here's a unit like this. You can see these spikes, right? So these are the spontaneous spikes, and this is where we're turning on the laser. Each laser pulse is only 5 milliseconds, and we apply this pulse at I think 20 hertz, pretty high frequency. But you can see that every laser pulse is associated with the spiking. So we think that whenever we see a neuron that responds so reliably, we're fairly sure that this is a neuron that expresses channel dopsin, right? And therefore, in this mouse, it's got to be a GABAergic neuron. So this is just other ways to show that these neurons are responding reliably. And once we identify a neuron like that, we record its activity over a long period of time. So here you can see is the EEG power spectrum, EMG amplitude. This is the computer automatic classification of brain state. And this is the firing rate of that neuron that I showed in the last slide. And you can see that this neuron shows a high firing rate whenever there's a REM, right? So here is always high. Now, during non-REM sleep, it's generally pretty low, and during wakefulness, it's actually sort of variable, and I'll come back to that point. But this is just the average firing rate within each brain state, and you can see that this unit has the highest firing rate during REM sleep. It's 26, 27 spikes per second. During non-REM, it's very low, it's less than 5, and during wake, it's intermediate. So this is important because in our optogenetic experiment, we applied the laser 20 hertz, right? So you can, to the first approximation, you can imagine we're basically adding 20 spikes per second. So the natural firing rate of this neuron during REM is 27 spikes per second. So that suggests that our stimulation protocol is not terribly non-physiological because we're adding, the number of spikes that's more or less sort of natural to their firing rate. So this is just one neuron, and this is a summary of 20 identified GABAergic neurons. And this is a little hard to see, but this is the same 20 neurons. For each one of them, we normalize its firing rate by its own maximum. And so you can see that all 20 neurons had the highest firing rate during REM sleep, right? So functionally, as far as sleep-wake modulation is concerned, these neurons are fairly homogeneous. They're all firing at maximum rate during REM sleep. So we also looked at the change in firing rate during transition. So this is the average firing rate of these neurons right at the onset of REM, right? So normally these animals would go from non-REM sleep to REM sleep. So this is the point when the transition occurs. And you can see that these neurons actually, their firing rates start creeping up about 20 seconds before the actual transition. And then they stay at high rate during REM sleep. And this is REM offset, right? So after REM, they usually wake up. And you can see that there's an abrupt decrease of firing rate at that. So the point is that there's an increase even before the transition, and the firing rate stays high during REM. So we think that this kind of firing pattern is ideally suited for both the induction of REM sleep and also the maintenance, right? Because if you want to induce REM, you got to start firing at high rate even before the transition happens. So this is the answer to the second question. So we know that these neurons are most active during REM, and also their firing rate change basically fit their functional roles that we think that they should perform. Okay, so the third question, what are their downstream targets, right? So we know that these neurons can induce REM sleep, and so how do they do it? So first we looked at the anatomy, and in fact we know from earlier studies, we know that the GABAergic neurons in the medulla have two targets. Some of them project anteriorly to the pons, some project down to the spinal cord. And when we looked at this anterior projection pathway, because we know that the pons is very important for REM sleep, we can see that, for example, here, this is the locus coeruleus. The red is tyrosine hydroxylase, which is a specific marker for the noradrenergic neurons in the locus coeruleus. And you can see quite a bit of green fiber sort of in and around this area. And in fact, when we did the rabies transsynaptic tracing, we've shown that indeed these neurons make direct synaptic connections with the LC neurons. So that's one interesting target. And the other target, at least we can see that the axon fibers, you see a lot of them in the VL pack, the ventrolateral periaqueductal gray. I always have a hard time with that word, but anyway, so VL pack. So the VL-PAG is interesting because there are earlier studies in which people showed that if you pharmacologically inactivate the VL-PAG by injecting mucimol, right, which is a GABA agonist, then the amount of REM sleep increases. So that suggests that the VL-PAG might be gating REM, right? So you have to suppress it in order for REM to happen. So now we see these GABAergic neurons projecting here, so we thought that this is interesting, right? Maybe this is how naturally you induce REM, because you can use the GABAergic neurons to suppress the VL-PAG. So what we did here was we looked at the VL-PAG neurons, right? So VL-PAG also has a lot of GABAergic neurons. And so if we express channel-rhodopsin in the VL-PAG GABAergic neurons and then turn on the light to see what they do to the brain, we saw, interestingly, a dramatic increase of non-REM sleep, a decrease of both wakefulness and complete suppression of REM sleep. So this is consistent with what people believed before using pharmacological manipulations, except that in addition to suppressing REM, it also decreases wakefulness, right? So it really increases non-REM sleep. So, because the other part of my lab is also really interested in looking for mechanisms that promote non-REM sleep. So this came as a pleasant surprise because this is what we're looking for, but this decrease of wakefulness is sort of an interesting side finding. Anyway, so we're pursuing this aspect. But anyway, so just to come back to REM, if we do closed loop stimulation, like we did for the VM neurons, we also saw a decrease of REM duration. So that really suggests that this VL-PAG GABAergic neurons suppress REM sleep. And then we did rabies virus tracing, right? So many of you are probably familiar with this technique. Basically, it allows you to trace, starting with the postsynaptic cell, and trace the monosynaptic inputs that directly innervate the cells that you're interested in. So here we targeted the VL-PAG GABAergic neurons. And for the sake of time, I won't go into the details because these methods are all established, published. But in any case, so this is the starting region. And we also see a lot of labeling in the ventral medulla. So VL-PAG and the VM are 2 millimeters apart, right? And yet the fact you see all these labeled neurons really shows that a lot of these neurons directly innervate the VL-PAG GABAergic neurons. And so we said, okay, so we know that these neurons, right, the neurons right here, they suppress REM. And we know that there's monosynaptic connection here. So the prediction is that if you just activate the axon terminals here, that should be sufficient to induce REM, right? Because the REM induction experiment before was to activate the cell bodies here. But then given the anatomy, our prediction is that you can just activate the axon terminals here, and that should be sufficient. And indeed, that's exactly what we found. So if we do the open loop stimulation, we saw the decrease of non-REM, increase of REM, and slow increase of wakefulness, pretty much exactly what we saw with the cell body stimulation. And also, if we do the closed loop stimulation, we saw this doubling of the REM duration, right? So pretty much we think that activating this axon terminal, if it's not accounting for all the effect of activating the cell body, it's really more than 90% of that effect. So we think that this pathway is really the main pathway doing that job. So to summarize this part of the third question, right? So we know that the VM GABAergic neurons innervate the REM suppressing VL-PAG neurons, and even just the activated axon terminals of VM neurons can induce and prolong REM sleep. So we think that the VL-PAG is an important postsynaptic target. It may not be the only one, but we think it's a very important one. And of course, this is only the beginning, and we're chasing down the other potential downstream targets. And now, question number 4, what are their inputs? So previous studies have shown that, so this is again a study done by the Lupi group in Lyon. And so again, they used CFOS, and they found, so basically these red dots represent the CFOS neurons that also project to the medulla, right? So it's CTB is a retrograde labeling dye, and then they also looked at CFOS. So basically, you do find quite a bit of neurons specifically in this region called SLD that are REM active because they're CFOS positive that also project to the VM. So we think that this is a very good candidate for the upstream region that might be driving the VM neurons to be active during REM. And so, so that was CFOS, and with low temporal resolutions, we wanted to take a closer look. And for this, we use this relatively new technique. It's a microendoscope green lens, because the SLD is buried very deep in the brain. You can't see it with your regular 2-photon on the surface. So we insert this green lens into that region, and then we attach this tiny camera developed by Mark Schnitzer's lab. So some people actually call it the Schnitzer scope. Anyway, so you can see that it's very small. This is important because even the mouse with a small body can carry it. And because we wanted the mouse to be sort of moving around in the cage, that's when they sleep the best, right? So the small camera is very important. So here's a video showing the glutamatergic neurons in or around the SLD region. So we're looking at calcium because we're expressing GCAM6 into the SLD glutamatergic neurons. So here, this mouse was basically moving around in his cage, and he can sleep or wake whenever he wants. This is the EEG. Right now, he's in non-REM sleep, and you can see some kind of flickering, especially if you look at these neurons, a little bit of the flickering during non-REM. But then you see soon he actually got into REM because of this theta, and you see that the whole field basically lights up, and then these neurons generally have much higher activity. So, okay, so just to summarize everything I told you so far, so I think that I won't read these. So these are the earlier part, but basically for the input part in the SLD, which we know projects the VM, that contains a population of REM-active glutamatergic neurons, the example I showed you in the last movie. And so here I'm sort of putting together this little circuit diagram of so far what we figured out in this part of the brain. So we know, so these are all color-coded. These light blue cyan neurons are the REM active and hopefully REM promoting neurons. So we show that there is a population in the ventral medulla. So we know that these neurons actually project to both the locus coeruleus, inhibiting the noradrenergic neurons. They also project to the VL pack, right, the GABAergic neurons, which I showed in previous slides. Now, if you look at the black neuron, which is a non-REM neuron, it's also GABAergic. We also know that they actually project to the SLD, and it seems like they actually innervate both the cyan neurons and the pink neurons, right? So, so these arrows are just the ones that we have figured out so far, but obviously there are a lot more connections. But it seems like so far, maybe it's partly wishful thinking, is sort of, but we think that the circuit is probably designed with some simple logic, which is that we have 3 brain states, right? Wakefulness, non-REM, and REM. Now, if you are a neuron that you want to promote a particular brain state, you gotta, and if you're GABAergic, you gotta inhibit the other 2, right? Because this blue neuron inhibits both the pink and the black inhibits both the pink and the light blue, right? So this is sort of so far what we have seen, and it seems like the scheme would make sense. But like I said, you know, there's a lot more to be mapped out in this region. Oh, the other thing is that I showed you that in the SLD region, there are these REM-active neurons. Unfortunately, there are also wake-active neurons. And spatially, they're pretty close to each other. So it's actually it's quite a challenge to try to separate these 2 populations of glutamatergic neurons. Okay, so that was the brainstem, and I'm almost running out of my time, and I'll try to move to the anterior part of the brain, and I'll try to speak faster. I think I'm already too fast, but anyway. So I'll quickly tell you about our study in the basal forebrain. Now, the basal forebrain, previous studies suggested that lesion causes, in some cases, insomnia. In other cases, it causes an increase in slow EEG activity, right? So this is a sleepier kind of brain state. So that suggests that the basal forebrain is important for both sleep and wakefulness. In terms of cell types, they're cholinergic neurons, so these are really well known because these are really damaged during in the early part of Alzheimer's disease. But there are also a lot of glutamatergic and GABAergic neurons. So we decided to take a closer look at each cell type. Now here is a recording, again using Optrode for cell type identification. So this is an identified cholinergic neuron. And so we follow this neuron over different brain states, and you can see that this neuron is active during both wakefulness and also REM, and completely shut down during non-REM sleep. Now you notice that the color scheme here is totally different from the earlier slides, and that's because these projects are done by different postdocs, and they have their favorite colors, and I could never convince them to change. But anyway, you just have to read the legend. So cholinergic neurons are like this, right? So this is the other notation. So on the horizontal axis, we're plotting the firing rate difference between the wake and non-REM state normalized by the sum. And the vertical axis is the firing rate difference between REM and non-REM. So if the dot, right, each circle is a single cell. So if the circle is in the upper right quadrant, it means that it's both REM and wake active. And you can see that all 12 cholinergic neurons are in the upper right quadrant, and they are wake and REM active. And in fact, this was known before. Barbara Jones in Canada, she actually recorded from these cholinergic neurons before. But we just wanted to sort of use a new technique to confirm what people have figured out before. Now, if we activate a lot of these cholinergic neurons, we saw decrease of non-REM sleep, increase of wakefulness, and not too much change in REM. So cholinergic neurons are wake promoting. And next, we looked at the glutamatergic neurons. So previously, people didn't know much about these guys. And then so here, I'm showing 31 identified glutamatergic neurons. And you can see that most of them are in the upper right quadrant again. There are only a couple in the other quadrants, but they're pretty close to the origin. So these are the cells that are weakly modulated. But most of them are really wake and REM active. And if we activate a lot of these glutamatergic neurons, again, we saw a decrease in non-REM, increase of wakefulness, no change in REM. Now, what about the GABAergic neurons? Now, we already knew, getting into this, that the GABAergic neurons are diverse. And so we knew that we had to somehow separate them. And the way to do that, since I came from the cortex, is to use these molecular markers. So in the cortex, it's very well known that there are these PV, parvalbumin positive neurons, and somatostatin positive neurons. And it turns out that both of them exist in basal forebrain, and they're largely separate. So first, the PV neurons. Again, most of them are in the upper right quadrant, a couple in the upper left. So these are the ones that are REM active, but not wake active. But when we activate these PV cells, again, a decrease of non-REM, increase of wakefulness. Right? So this is just basically confirming what I told you at the very beginning. It's really easy to find neurons that promote wakefulness. Right? So 3 out of the 4 cell types. But fortunately, when we moved to somatostatin neurons, we were bored to death. Right? It's always wake, wake, wake. But finally, when we went to the somatostatin neuron, we saw some neurons are actually sleep active, right? So here you see that this neuron is completely shut down during gray, which is wake, but it's really active during non-REM, during this white region, and REM, yellow, is kind of intermediate. And when we plotted all of these neurons, right, you see that it's all over the place. Unfortunately, we also see some wake and REM active neurons, but we also saw some REM active neurons, some non-REM active neurons, and also some in the 4th quadrant. Basically, they're everywhere, right? So somatostatin, unfortunately, is a very diverse population. But at least this is the only population that we see neurons way out here. So these are really the non-REM active neurons. And when we activate the entire population, we actually finally saw we can promote non-REM sleep a little bit, decrease the wakefulness a little bit. Right? So basically what's going on is that it's a diverse population, but the overall effect is to promote non-REM sleep. So these are the functional characterization of the 4 cell types. And then, of course, we're also interested in the circuit. So one of the things we did was to look at the local circuit within the basal forebrain. So this just illustrates our technique. We would express channel rhodopsin in one cell type. In this case, it's cholinergic. And then fluorescent marker, TG tomato, in another cell type. In this case, is PV. And then we patch on the red neurons and turn on the light to activate the green neurons and look at the synaptic connection. So this is our data, right? I don't expect you to look through all of them. But basically what it is that there are 4 panels, right? So each one shows one presynaptic cell type. And within each panel, there are 3 boxes. And that shows each of the 3 postsynaptic cell types, right? So we're basically mapping all the pairwise connections, really a lot of work. And here I'll just summarize what we found. So these are the 3 wake-promoting neurons. I'm using a light circle to represent them. This is the one cell type that promotes non-REM sleep. So what we found is that the wake-promoting neurons seem to be organized in a hierarchical chain with the glutamatergic neurons at the top, because these guys actually excite both the cholinergic neurons and also the PV neurons. Now, the cholinergic neurons, interestingly, excite the PV neurons, but they mostly inhibit the V-glut neurons, right? So that's why I'm saying it's sort of, there's a hierarchy, because the feedback is either inhibitory, and in this case, we could never detect any PV to cholinergic connection. So, but the more important thing here is that the one type of sleep-promoting neuron, the somatostatin neurons, actually provide broad inhibition to all 3 types of wake-promoting neurons. And in fact, this is what we see in the other brain regions as well, in the sense that if you want to promote sleep, you better broadly inhibit a lot of the wake-promoting neurons. Okay, so at this point, right, so we found somatostatin, finally they promote sleep, great, except that we know it's a diverse population, right? So what do? We can keep trying other markers, right? I have a very long list of candidate markers, but trying each marker takes several months, right? So my postdocs were just not interested in doing that, right? And so, I mean, believe me, I tried both coercion and bribing and all of that, but nothing was working, right? And so, so we decided to take a different approach, which is, inspired by this finding that the sleep neurons actually inhibit the wake neurons. So we thought that could we use that feature to try to hunt down the sleep neurons based on the fact that they might want to innervate the wake-promoting neurons if they're GABAergic. So, so this was done by Shinjae Chung, a really talented postdoc in my lab. So what we did here was that we injected the retrograde tracer, retrobeats, into a whole bunch of known postsynaptic targets. These are the regions that we know promote wakefulness. One is the TMN, so this contains the histaminergic neurons, which are very important for wakefulness. DMH, also in the hypothalamus, and the VL-PAC. And we also did a CFOS staining after sleep rebound. And so what we found is that it's the neurons that are retrogradely labeled from TMN, right, that occupy the highest percentage of the CFOS positive neurons, right? In other words, if we look at all the sleep active neurons based on CFOS staining, we see a lot of them projecting to the TMN. So that suggests that the TMN might be an interesting target to go for. So then we collaborated with Ali Setin. So Ali, when he was a postdoc in Ed Calloway's lab, developed this really cool lentivirus tool. It's pseudotyped with rabies virus glycoprotein, and that makes the lentivirus travel retrogradely. So normally lentivirus doesn't travel backwards, but once you pseudotype them, they do. So what we did here was to inject the lentivirus in the TMN region, right? This is the target we decided to focus on, so that the virus can travel backwards to the preoptic area. And then this virus expresses channel dobson YFP in a Cree-dependent manner. So if we inject this into the GAD2 Cree mice, then we can label the GABAergic neurons, the subset of them that projects to the TMN, right? So it has to be cell type, but also projection specific. And so, and then we did CFOS staining, right, for sleep active neurons, and you can see a lot of overlap. So that suggests that this strategy might be a useful strategy. And so, so we activated those neurons, right, the subset of GABAergic neurons in the preoptic area, which is very close to the basal forebrain. And so you can see that these are the 2 trials, right? So in one trial, the animal was awake. We turn on the laser, here, this blue shading, and quickly the mouse got into non-REM sleep. Here's another trial. The mouse was in non-REM sleep, and after we turn on the laser, the mouse got into REM sleep. So these are the 2 trials. But here's a summary of the probability, right? So you can see that we turn on the laser, right? And that, as soon as we turn on the laser, there's a very sharp increase of the probability of non-REM sleep. Also increase of REM, but a decrease of wakefulness that stayed low during this 2-minute period that we turn on the laser, right? So this, it seems like this subset is promoting both REM and non-REM sleep. Now this is in stark contrast to the effect of activating all the GABAergic neurons in the preoptic area. So if we just inject the AAV into that area, into the GAD-Cree mouse, then we saw this dramatic increase of wakefulness, right? This is what I told you before, right? If you just randomly do it, you wake up the animal. But since this is a subset of the GABAergic neurons, right, it's got the opposite effect. So we think that we're really using this method with a pretty effective method to sort of sort out the minority of sleep-promoting neurons in the preoptic region. Now, if we do this for the glutamatergic neurons that project to the TMN, again using lentivirus, again we saw promotion of wakefulness, right? So this suggests that you have to, if you want to get sleep, you have to not only target, it has to be cell type specific and also projection specific. If you lose either one, then you basically get wakefulness. Okay, and then we did recording, right? Again, optogenetic tagging and recording, right? This is just to show that we identified a GABAergic neuron projecting the TMN using this optogenetic method. And then here's the recorded firing rate. Here is just one example. And this is a summary of, I think, 17 identified neurons, right? The GABAergic projecting the TMN. And you can sort of get an idea that they're really active during REM and intermediate during non-REM, but they're least active during wake, right? So this is kind of what we're hoping for. Now this is another, using this two-dimensional scatter plot, except in this case we change the axes so that on the right it means that it's more active during non-REM, up is more active during REM, right? So the blue dots are all the identified GABAergic neurons that project to the TMN, and you can see that most of them are in the upper right quadrant, so they're non-REM and REM active. So this is different from the randomly collected GABAergic neurons in that region, right? So they're all over the place, right? So some of them are wake active. So again, this experiment suggests that using this retrograde labeling, we're sort of sorting out a subset of the neurons that are sleep active and also sleep promoting. Okay, so here's a quick summary, right? And I won't read it. I think I'm really running out of time. So last bit, right? So far we're using this lentivirus, and it's really great because without that we couldn't possibly have sorted out the POA neurons that promote sleep. But lentivirus is also not that common, right? So every time we want to do something different, we have to beg Ali to make a different construct for us, and we rely on the Salk Institute viral core, and they're not terribly reliable. I hate to badmouth them, but so far, the last few months, we hadn't had a great experience. So we would rather use AAV, which is, everybody uses them, and, they're very reliable. But how do we do that, right? So AAV is not retrograde. And so the way to do that is that we got to find a molecular marker so that we can use a Cree line, because once you have a Cree line, you can just use the Cree-dependent AAV, right? So you go back to the easier method. But how do we find a marker? So for this, we did gene profiling, and this is a single-cell RNA-seq in collaboration with the Allen Institute, in particular, Basilka, Tuk, and Honghui Zheng. So what we did was to back-label the neurons that project to the TMN, and we dissociate them when we did single-cell RNA-seq. And when we say we, actually, really, they did all of this, because in my lab, we didn't have the expertise. And this is what the data looks like. Each column represents so each row represents a single gene, and each column represents a single cell. So in this batch, they sequenced about 70 cells for us. And so if you see, a really red row, that means that that particular gene is heavily expressed in the majority of the cells that we have sorted out, right? So we found that one of them is called TAC1, tachykinin-1. So this is a gene that encodes this neuropeptide substance P. And here's another one. This is actually prodynorphin. So it turns out that TAC1, there's already a Cree mouse line sold by the Jackson Labs. So we decided to test that. And so, and again, because I'm running out of time, I just quickly go through. So basically, this is showing that these TAC1 neurons are, they do overlap. The TAC1 marker does overlap with the retrogradely labeled cells that project to the TMN. And this shows that if we express channelrhodopsin YAP in the TAC1 neurons, and if we look at their axon fibers in the TMN region, we do see them, right? So both experiments just show that, yes, these cells do project to that region. So our single-cell RNA-seq wasn't giving us some really spurious effects. Now here's a key experiment. So what if we just express AAV with channelrhodopsin in the TAC1 neurons in the preoptic area? And when we turn on the laser, we saw an increase of non-REM, decrease of wakefulness, not too much change in REM. At least we're promoting sleep. So single-cell RNA-seq is one method. The other method we use is the so-called TRAP method, translating ribosome affinity purification method. It's basically the idea is to inject this virus, right? This is also retrograde, but we're labeling the cells that project to a particular target with a ribosomal protein L10A that has the H8 tag. And then we grind out the tissue and then use the antibody against HA. And this antibody is attached to the magnetic beads so that we can pull down the ribosome, right? And then because the ribosome is doing the job attached to the mRNA, we can pull down the mRNA together with that. And then we can sequence the mRNA. And so this is what the data looks like. Each dot is a single gene. We're plotting the expression level of this retrogradely labeled population against the entire population. So if you see a dot above the diagonal, that means that this gene is more heavily expressed in that retrograde population. So among them, we focus on two genes. One is CRH, the other CCK. Both are neuropeptides. And there's some previous literature suggesting that they might be involved in sleep regulation. And so again, because they also have the Cre lines, so we can test them. So again, these are just the anatomy to show that these neurons do project to the TMN. But here's the key experiment. When we activate the CCK neurons, right, you see this increase of non-REM, also increase of REM, a dramatic decrease of wakefulness, and this is the effect for CRH, right? So smaller magnitude, but at least in the same direction. So we think that basically using this gene profiling is a much more principled way to sort of pull out the markers that might label a subset of the GABAergic neurons that really promote sleep. And so that's where we are. Obviously, there's a lot more work to be done, but this is sort of just a progress report. Anyway, so these are the people in my lab who did the work. Franz Weber did the study on the medulla neurons controlling REM sleep. Julie is the one who got the endoscope imaging to work. Minxu and Xinjie are the two pioneers in my lab. They were really the first people who ventured into sleep research 5 years ago. Minxu developed a lot of the key techniques, including opto recording in my lab. And Xinjie is the person responsible for this gene profiling project that I talked about in the last part. Thank you very much. Okay, so are there any questions?
This is on? Yes. Hi. I'll ask a question about just the first part, the medulla. Basically, so you showed that the stimulation of the projections from the medulla to the VL-PAG induces REM, and then separately you showed that stimulating the VL-PAG itself has the opposite effect. Have you done a functional occlusion experiment? Because they were both done in the same mouse line, in the GAD line. So could you put channel rhodopsin into both the upstream in the medulla and in the VL-PAG and see if you can occlude the effect of stimulating just that projection? Have you done that experiment?
We haven't done that, but I think that would be an interesting experiment, right? So you just inject the virus both in the VM and also in the VL-PAG, and you just stick an optic fiber
over the
in the VL-PAG and see, right? I guess so what you hope to see is to compare that with just the VM or just the VL-PAG, right?
And if it's truly
I think it's totally
downstream,
doable, except that you have to. It's a between subject comparison, right? So that would be, I think, you just need more data to really be sure about that.
Sure.
But I think it's a meaningful experiment.
I was wondering if you could explain a little bit more for the second part of your talk. How do you decide what frequency to stimulate those neurons that you're profiling to see what their effects are for a program or?
So, ideally what you want to do is to first do optical recording and figure out their natural firing rate and then set your laser frequency according to that. But in reality, optical recording is a very labor-intensive technique, right? Just to be honest, I think that our current success rate depends, right? The different cell types, if it's a very sparse cell type, it's even harder. But I would say that average everything is you probably get a cell once every 3 days if you're good. Right? So usually it takes a long time to really get that information. So my attitude is that initially you just want to use a relatively low frequency. If you can see the effect, I mean, you're safer, right? Because it's more likely to be within the physiological range. But if you use too high, I normally would never go up to 30 hertz because I think that then your probability of being non-physiological is much higher.
Really nice stuff.
Thank you.
It looked like if I understand this correctly, then the GABAergic neurons were suppressing the wakefulness neurons. And the question is it really GABA that's doing it? Because as you pointed out, there's often a lot of peptides co-expressed with that. So do you have any evidence of that yet? Or?
So that's a really, really good question. At this point, we're really sort of in our first order business, right? Just to try to find a marker and see who are these sleep neurons? Can we use the marker to reliably hit them and get reproducible effect? But the downstream mechanism, there's a lot more to be done, right? Firstly, these markers that we happen to pick are the peptide markers, and that immediately opens the question, is it GABA or is it a peptide, right? So I think that those are really important questions. And in the end, my guess is that the answer is both.
Join me in thanking Dr. Dan. And I want to acknowledge Dr. Gottesman, who's down in the front and join us, and also invite you to a reception in the NIH library right immediately following the lecture. Dr. Dan will be available to answer more questions there. Thank you for coming.